stop doing that? That's where that went. Get out. Push. I'm going to quickly say hello to the class. Hey, hey. There you go. You know how to say something?
Yeah, puppy issues. God damn it, dog. He's got this habit of putting his ball underneath the couch that I'm sitting on. Give it here. Here. There you go. And then yowling at it until I get it out from him again. That's why I'm a little delayed. Ah, right. Let's get rid of that. Oh, where are we? A little less organized than I should be. Because of that little peril. Okay. So we finished up non-Mendelian genetics, I think, from what I can remember. Is that right? Or did we? Maybe we didn't.
We ended with the Can Dynasty, the map. I think that was like lecture 7.
Oh, okay. Shit, maybe we didn't. I'm sorry. I'm kind of all out of sorts at the moment because of that invited lecture and the whole Snow Week thing. Oh, yeah. No. Yes. Oh, no. Yes. Fly. Right. Thank you. Okay. All right. At least a little bit is behaving himself. Okay. So. Don't need to look at the schedule. So actually, oh, the Genghis Khan one. That's right. Yeah, I got you. Okay, so that was the whole Y chromosome haplotype deal with Genghis Khan, right? Okay, awesome. I know where I'm at now. Okay, so what we have to do today. Actually, how much time do we have for linkage? Let me just double check. Oh, we got a couple of good lectures on that. Okay, because that's a real doozy of a topic to talk about. Okay, so don't need this. While that's open, do need this. Right, so essentially, what we're talking about on, so the previous part of non-Mendelian genetics was really about sex determination, sex linkage, right? So the differential expressivity of traits depending on sex. So whether you display the phenotype, oh God, it's done it again. Whether you display the phenotype or not depends on whether you're a male or a female, essentially. And so that almost always relates to genes on sex chromosomes. And we've covered epistasis as well. We should have, I think. Have we? Oh no, that's today. Right, okay. Well, I do know what I'm doing, honest. All right, so kind of what's left are talking about dominance, right? What that actually means. Oh, you got to excuse me for a second. Is say hello to the class, you little pest. See. Never have these problems when I'm teaching in the classroom. See, it just brings it back. Yeah, aren't you clever? Now you stick it under the table in about 30 seconds. All right, so where was I? This is like kind of you know, boot camp training for professors, how to teach in a distracting environment. Anyway, so there's the case of dominance, right? We've talked a lot about dominance in fairly simple terms so far. We need to talk about how that varies, right? It's not an absolute. It kind of depends really very much on biochemical function. Oh shit, it's done it again. I need to get my wife down here to keep an eye on him. It really depends on what's going on at the molecular level, right? And so picture you have in front of you is of snapdragon flowers. And you can see that essentially we have an intermediate form, right? So this kind of different dominance called incomplete dominance is often a pigmentation issue, right? And we'll talk about more about that in a little bit. We need to talk about dominance of one trait over another, right? So not just dominance and the allelic level, you know, individual alleles of a gene, but also in terms of how function at one locus or the phenotype you get for one trait affects the expression of the phenotype for another trait, right? And there are several cool examples of that we can talk about. That's called epistasis. What, you want to talk to them? He just wants to chew. Okay. So that's epistasis, right? And really what we're trying to get at with a lot of this non-Mendelian genetic stuff is really a bigger picture, right? So Mendelian genetics is really, you know, the whole monohybrid, dihybrid cross. It's pretty simple stuff, right? It's typically all about one gene, two alleles, two phenotypes, right? Or one trait, one gene, two alleles, two phenotypes. And how those are inherited and expressed is fairly simple. Now, obviously, we're not that simple, right? Even peas aren't that simple. There's a lot more to peas than just what Mendel discovered. And so all of this really kind of fills in that huge space that Mendel didn't really go into. Largely because it's just too hard, or it was too hard for him then. It's still hard now, right? And so there's epistasis, right, which will kind of make be fairly intuitive once I kind of go through the ideas behind it. Get out of it by my hand now. There's also the issue of kind of complex traits, right? So almost all human traits, almost all, not all of them, but almost all of them are not caused by one gene. There are some disease traits, particularly congenital diseases, which are caused by one gene. Cystic fibrosis is a classic example, right? Or sickle cell disease is another one, right? That's actually another example of something else. But even with cystic fibrosis, right, you have one gene. Last count, there's something like 1,500 different alleles for cystic fibrosis that can cause that disease if inherited in the right pattern, right? So even for single gene traits, it's not simple, right? And most traits are not single gene traits. You know, for example, out of it. Give me just a moment, would you? All right, puppy problem has been solved. At least it's now my neighbor's problem and not mine. Okay. All right, I can think straight now. He's very cute, but Jesus Christ, is a complete pain in the ass. Anyway, so epistasis. Oh yeah, complex traits, right? So, um, kind of fairly obviously, right? Most human traits are complex, right? They're a result of multiple steps in the biochemical pathway, like metabolism, for example. They're a result of multiple time coordinated events, such as development. Why are we the height we are? Why am I taller than other people and shorter than some? It's because of a different collection of alleles that I have in different genes, which control many different processes. How long does my leg development go on for, right? How different changes in bone development, different changes in muscle development, right? Different strengths of signaling from, what will become my tootsies to the rest of my leg. All of those things are controlled by different genes, right? And on top of that, you also have the environment as well. The whole, essentially, it's kind of boiled down to the whole nurture versus nature kind of deal. And if you had kind of, plonk me down in war-torn Yemen or Syria, maybe, where there is severe malnutrition, particularly in Yemen, it's an absolute disaster there. Almost certainly, I would not have grown to the height that I did growing up in the UK, because my nutritional environment would be different. I would be under considerably more stress, right, than I would be growing up in rural England, which wasn't really that stressful thinking about it. And that the effect of the environment would have interacted with the collection of alleles I had for different genes controlling that trait, and I probably would have ended up quite a lot shorter, right? So there's that kind of issue as well. Hey, love, little bits outside because it's been a complete pest. I can't teach. So kind of really essentially what we're trying to do in the space of like 2 lectures is give you like the much bigger picture of genetic complexity in terms of inheritance, and it is phenomenally complex, right? If you think about your own family tree from perspective inheritance, right, there will be some traits which pop out as kind of being family traits, right, whatever those might be, and they will pass down through generations and you can follow them, right? But for the rest of it, it's a big question of like, well, why am I the way that I am? How did I end up this particular with this particular phenotype? So even with all that we know about yeah, all that we know about genetics now, we still really don't understand an awful lot of why we are the way we are, just because it's so complex, right? There are so many different interactions which are very, very hard to figure out. Okay, so that's kind of like the big picture stuff, right? In terms of detail, we're going to deal with dominance first. And really, dominance is, it's kind of making a statement about function, typically, right? And so in terms of dominance, really what we're talking about is something working properly, right? And there being enough of that something that you only need one functional copy of that gene to provide function, right? So if you're producing an enzyme for, I don't know, TCA cycle, let's say, one gene should be able to produce enough enzyme for you to not notice any difference in terms of function, right? Because that's what we care about. Is this thing working or not? If it works, it's normal. If it doesn't work, then something's up, right? And so even if you have a non-functioning copy of that gene, it doesn't matter, right? That copy might not even be there, might not even exist, right? It might be a huge gaping like crater in the chromosome where that gene should be. And it doesn't matter because you're providing the function of that gene from just your one copy. That's typically what dominance means, right? And so as long as you have a functioning copy, you have function, right? Now there are some cases, and there aren't very many of these, right? They're actually really hard to find. There are some cases where that dominance doesn't quite work that way, right? And those cases are incomplete dominance and codominance. I'm going to need to use my whiteboard in a second. And they have particular characteristics, right, by which you can recognize which one you have and whether it's departing from the classic dominance recessive kind of format. Let's put this back how it was. Here we go. All right, and so first one is incomplete dominance, right? And so and essentially what that's saying is that the function of one copy of the gene is insufficient to provide, oops, here we go. To provide full function. And so these are traits which are typically pigment involved. Right, and so typically if you have no pigment production, you have a lack of color. In flowers, that's white. In mammals, that's typically kind of yellow, kind of yellowy ginger kind of color, right? Is the classic absence of color. And in incomplete dominance, you need both copies functioning. Two full functioning copies of the gene or genes, right, that produce pigment to be able to mask that recessive white phenotype. One is not sufficient, right? It's actually a term for this called haploinsufficiency, which you don't need to worry about, but it's kind of a neat word. And it's very much like paint, if you think about it, right? You know, if you're trying to paint over, you know, a color over a white wall, typically you need more than one coat to be able to get complete coverage, right? So you don't see any of that underlying white color there bleeding through, right? And so there are two kind of characteristics of incomplete dominance. One is that you have an intermediate phenotype for the heterozygote. So in kind of classic dominance, the heterozygote will always show the phenotype of the homozygous dominant genotype, right? Because you just need the one allele to be able to have full function and have that phenotype. In incomplete dominance, one is not sufficient, right? And so you have this intermediate phenotype for the heterozygotes. And second thing, oops, in a monohybrid cross, the F2 generation is a 1:2:1 phenotype. Oops, can't see what I'm writing. Ratio. Right, so if you do a cross with snapdragons, and we've got, here we go. Go to down here, you'll see that if you cross two heterozygotes together, which have an intermediate phenotype, neither of their parents, but somewhere in between, then you will get a 1:2 ratio of red to pink to white or ivory flowers, right? That is the classic, those two things together is the classic incomplete dominance inheritance, I guess. And actually, my little research is coming downstairs. You see that for leopard geckos too. So leopard geckos have a crazy array of different kind of patterns and pigmentation and stuff like that. And with a trait called Mac Snow, you have incomplete dominance where the heterozygotes have an intermediate level of dark pigmentation compared to the homozygous recessive, which has very little, and the homozygous dominant, which has a lot and also has black eyes as well, which the het does not. So that's kind of incomplete dominance. It's a pretty straightforward thing to think about. And codominance is very different. So typically you can only express one phenotype at a time, right? You can either be, red or pink or white or have a disease or not have a disease, right? In the case of codominance, there you go. And this is also very rare too. It's about, did I get you, little bastard? Expressing two phenotypes at the same time. Now, this is an even harder one to find examples of. So the example in the PowerPoint is about the kind of the molecular biology, essentially, of wrinkled versus round peas, which is a good one, right? So now you're looking at the phenotype in terms of its gel electrophoresis pattern, right? But I think a better example is red blood cells, right? So this is one of my fave ones to talk about. And trust me, I've literally spent years, ever since I started teaching this, trying to find other examples to talk about. And this year I managed to find the one about leopard geckos, which is cool. From going to a reptile fair, I've never found another example of codominance in terms of things that you can see. Okay. So I'm gonna, there we go. My whiteboard up. So in red blood cells, you have 3 different alleles. A, B, and O. These are dominant over O. Right, so you can have, hang on, let's not go there just right now. Okay, so essentially what these are involved in are antigen production. It's my little shorthand for production. So if you have A, you have, that's supposed to be a red blood cell. It's not a donut with a marshmallow stuck in it. You have one particular type of antigen on the surface of that cell. I don't know if it's a different kind of glycoprotein or what. I'll need to look into that. Always more things to learn. If you have the B, you have a different shape, right? Actually, I really do need to look into this because I should know more about the molecular basis of those antigens. My guess would be a glycoprotein. That's typically the kind of antigens that we produce. And O is no antigens. Make sense so far? Okay. So if you are, and this is actually cool because it's something about your own biology. I'm B positive blood, which is kind of neat. There are some people that are O, which is pretty awesome, and obviously others. Right. So if this kind of can follow the same kind of classic Mendelian genetics, right? So you can be AA or AO, and that gets you the A antigen, right? AG for short. You can be BB or BO equals B antigen, and OO equals no antigen. Right. So far, so simple. And if whatever, and just kind of you don't need to know this for understanding codominance, but it's fun to understand blood type and blood doning. Whatever antigen you produce, you don't produce the antibody to it, right? Because obviously otherwise you'll identify your own blood as being foreign and you'd attack it, which would be generally a bad idea, right? So let's move this over a little bit. I did this on my whiteboard in my office for the other class, which was absolutely fabulous. Hang on, let's do it in a different color. That'll make it a little easier. There we go. So you get B antibody. If you're A, you get A antibody if you're B, and you get both if you are O. Right, so that's why O's are the universal donors, because you strip all the other gumph out when you prepare blood for transfusion, right? You don't include any of the plasma with it. So all the antibodies and stuff go away. And so O is essentially immunologically inert, right? It doesn't have any antigens, so you can give it to anybody and they will not react to it. Does that make sense? You can only give A blood to people that have A blood or are the one that we're going to talk about in a little bit, which is AB, right? So that's why O is a universal donor. This is why it gets all kind of complicated. That's why these people are very, very highly sought after blood donating services. Now, if you are, and stop me if you have.
Yes, I just had a question regarding because like I am O negative. So what is the RH?
Oh, the rhesus factor? I've no idea.
Because the RH is like I was negative for the RH, so that's why it's like O negative.
Right.
But what does it have to like play in?
The rhesus factor is something that's a maternal trait. I don't know. I'll look it up and I'll let you know. But it's one of those things where I keep meaning to look it up and I keep forgetting, basically. But it's something that's completely different from the antigen that you have on your red blood cells. It's something else. I just don't know what that something else is.
Okay.
And to Rose, that's 100% correct. You is the universal donor, but you can only, if you're O blood type, you can only receive blood from, it would be other O people. Is that right? Yes, you can only receive blood from other people that are O. You can't receive it from any other type of blood donor. So it's great for everybody else. It kind of sucks for you if you're O. Sorry, what did you were O, weren't you, Tanya? Oops, shy. Shouldn't have said that.
Yes.
So you're great for the community. It's much less ideal if you're actually O for your own biology. Now, the difference is if you're, so these are heterozygotes with a recessive allele, also heterozygote with a recessive allele. So you show the phenotype of the dominant allele. Now, let's make sure you can all see this. You can have AB, right? So this is two dominant alleles together, right? And so now what you get is A and B antigens, right? So it'd look like cartoon style, like that, basically, right? Both antigens will be present on the surface of the red blood cells. And as a result, people who are AB have no antibodies. So AB individuals are universal. I can't fit recipients in there, can I? There you go, sort of tiny bad writing. Are universal recipients. Let's move that over there. So now you have 3 alleles for one gene. You have 1, 2, 3, 4, 5, 6 different genotypes and 4 different phenotype, well, 4 different phenotype combinations essentially. Because both of these, the phenotypes from both of these dominant alleles can be expressed at the same time. And that's what codominance is all about. Yeah. And that's why it's such a pain to find good examples of that, right? Because there are very, very few of them. Anybody have any questions? Okay. And also, this is something that you don't tend to come across. The reason why we talk about this is really to illustrate how dominance is not a fixed property, right? It very much depends on the function provided by that gene, whether you need 2 copies of that gene to function in order to have full function for that particular trait, and also the context of that particular trait. You know, so in this case of red blood cells, you know, if you had other alleles, right, that produced other antigens, you'd have an even more complex situation, right? Because all of those can be present at the same time. And so really the limit on how many you can have present at the same time is down to how many copies of the gene you have, right? Which is always 2, right? So if a new allele for red blood antigens emerge, oh, just saw your question, Rose, I'll get to you in a second. Then potentially you'd have a new one, which could be co-dominant with the others at the same time, but you could only ever express 2 different phenotypes at the same time, simply because that's the number of copies that you have. Hang on, let me just look it up. Go on, Wikipedia, where are you? Dum, 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 dum. So it's another antigen. God dang it. That just makes things even more complicated. Yeah, I'm not going to be able to summarize that in any kind of coherent fashion just by reading it. I'll have to do some more research on that. Sorry. Oh, that's actually, I had that answer from my question from my daughter too. So, no. So what you see with intermediate kind of mottling is it's either incomplete dominance or it's something called mosaicism, right? So that's where the gene controlling pigment production or import or whatever, whatever it is that controls whether the fur is black or white, that's switched on or off in different kind of clumps of cells, right? So the classic example of that would be, oh dang, what's it called in cats? Calico, that's it. It's called mosaicism. Mosaicism. And that's because in cats, essentially it depends on particularly in female cats, which X chromosome is turned off in which kind of pigment producing cells. So in your example, Rose, codominance would be crossing white and black cows, and each hair, is back again. Got to let in. Each hair would be both black and white. And that's obviously not something that can happen for pigment. You can only have one color, whatever that color might be. So no, that would not be codominance. It's actually, trust me, it's astonishingly hard to think of examples of codominance. Quite often when you think of codominance, what you're actually thinking about is organism-wide changes in expression of a trait, right? And so kind of patterning, particularly when it comes to pigment, is due to that. Some different causes in different contexts, right? Siamese cats, it's about temperature sensitive mutation. Shush. Sorry, talking to the dog. Well, he's kind of being quiet right now, so maybe he'll not be a pest. So in Siamese cats, if you have, I think if they're white at their extremities, then the gene gets turned off. If they're darker at the extremities, the gene gets turned on when they're cooler. And because, your extremities are a little bit cooler than your core, that's what gives that pattern. Other ones, it's whether a gene is turned on or off somewhat randomly. That's how you get those different patterns. It's actually very complex. I don't fully understand it in cats. And cats are probably the best known example of that kind of thing. There are lots of different genes interacting. So that's relatively straightforward in the grand scheme of things. Oops, where are we at? However, what is really interesting to talk about is epistasis. Let me I've taken a picture already. Let me wipe off. Board. Right, and so epistasis also has a bunch of characteristics, right? It has particular things that you would expect to see depending on what kind of epistasis is occurring. And essentially, really what we're talking about with epistasis is You remember like the whole law of independent assortment, right? What alleles you get for one gene have no effect of what alleles you get for another gene, right? And so those two traits should behave perfectly independently, right? Well, there's two cases where they don't. One is epistasis, the other one is linkage, right? Linkage is based on a physical interaction between alleles of different genes based on their proximity. Epistasis is based on a functional interaction between what those different loci do, what those different genes do, right? And essentially, in kind of in a nutshell, it means that the phenotype you have at one locus for one trait affects the expression of the phenotype from another trait. Okay, and so the easiest way to talk about this is with recessive epistasis. There's a lot of kind of board work here. He's busy chewing on a bone, so hopefully he'll keep us in peace for a little while. Right, so essentially recessive epistasis is typically about biochemical pathways, right? So even something as straightforward as pigment, right? Why you have brown hair or black hair or otherwise is the result of multiple steps in a biochemical pathway, right? And so if it's something just as simple as this, right, we have enzyme 1 and enzyme 2, right? Whatever this might be, this is your precursor, this is your C would be your final product, with B being an intermediate. You need both of those functioning in order to be able to get your final product. So in this case, the phenotype that you're looking at here is enzymatic activity or functional production of the next step, right? So enzyme 1's phenotype is production of B, enzyme 2's phenotype is production of C. Does that make sense? That's kind of what they do. That's their job, essentially. And so what would happen to the phenotype of enzyme 2 if you have a homozygous recessive genotype for enzyme 1? This is not, codependence. Yeah, I guess that's one way of talking about it. We call it epistasis because we're actually talking about the order in which things kind of occur as well. So what would happen to the phenotype of enzyme 2? Let's say you have AA and genotype here, and we have homozygous recessive genotype here. What's going to happen to the phenotype of enzyme 2? Anybody? So if enzyme 2 is all about taking B and making C, what happens when you get rid of enzyme 1 function? You get a change for sure. So let's put it another way. So the dominant phenotype would be producing C. And if I should have done it the other way around, shouldn't I really? Right, so that's what you get, let's say, in a, if you have, actually, don't even really need that one there, to be honest, right? This is a classic dominant trait, dominant recessive trait. So enzyme 1 is all about producing B, enzyme 2 is all about producing C, right? The output of both of these together produces C, right? Now, if you have a, let's say, hang on, let's do it. If you have homozygous recessive A, right, you don't get any C, right? Because this function doesn't happen anymore. Does that make sense? It's kind of like classic Mendelian stuff. What phenotype do you get for this gene if you're homozygous recessive for this one? Do you get C or do you not get C?
So if it's recessive, no C, and if it's dominant, you will get C?
For this one, correct. For this one too, right? So if both of these function, right, you get C. If either of these does not function, you don't get C, right? So both of these are required for you to see the phenotype of, let's get a different color, of this genotype, right? Exactly. You don't get B because you don't get C because you don't get B, right? So you need the function of this earlier step of this different genetic locus, right? This is encoded by a different gene entirely, in order to see the phenotype of the dominant allele in the second step. So remember, I'm teaching, okay, love? And so what genotype you have for this locus affects the expression of the phenotype from this locus. Right? These two loci, these two genes, are not independent. The expression of their phenotype is not independent. One relies on the other one. Oh, okay. Yeah, that's a really good question, Rose. That was what I was getting to next. And so if that makes your head spin, let's look at, so actually, hang on, before I do that. So essentially what you're going to get from this, if you did a dihybrid cross, right? So we've got two genes. Each gene has two alleles, right? So just the same as if you were doing a dihybrid cross. Now, in the parental generation, you'll have only two phenotypes. right? You'll have, hang on, just wait for a second. There we go. Let me just bring up the, come on. Right, so this is recessive epistasis, right? So it's essentially what I've drawn out on the board. If you do a dihybrid cross with these two genes, you'll get, well, this is a kind of in trans. You could cross either a pigment flower with a non-pigment flower or two non-pigmented flowers, and you still have the same combination of alleles. You'd have the same F1, right? So in the F2, if you cross two heterozygotes for both genes, right, you're going to have pigmented flowers in this case, right? You're going to see the product of that pathway, purple in this case, right? So they're heterozygotes. They have function for both of those loci because they have one dominant allele for each. However, when you cross those together, you don't see a 9:3 to 3:1 ratio. You'll only see two phenotype combinations or just phenotypes. One, pigment or function, essentially, of the pathway. Second one, no function, right? And so instead of a 9:3 to 3 ratio, now you see a 9:7 ratio, right? Because all of these white ones are ones in which they are homozygous recessive for one of the genes. The one down here is that homozygous recessive for both of them. That's typically the one that you see in the dihybrid cross with the recessive phenotypes. But now you have to have a dominant allele in both, and so those 3 in the 3 now show the recessive phenotype. That's recessive homeostasis. I'm just going to show you a real quick one. Anybody have any questions about that before I move on? Okay. You can also have, and this, I'm just going to do this quickly because it's not critical to understand the process. It's just kind of a fun one to talk about. Now, there's my other color. Hey, here it is. You're going to have to use a little bit of imagination because A, I can't draw white cats on a whiteboard and you see them. It'd be like a metaphysical cat, I guess. And also, I don't have colors that correspond to colors that you actually find in cats. So, god damn, these things piss me off no end. Isn't that a cute cat? I'm not quite like Simon's cat level, but kind of close enough. Okay, so if you cross, actually, let's use a different color. I'm going to use, this is supposed to be like a ginger cat. Right. Essentially, you can cross, this will be, so this is about dominant epistasis. This is the white gene. Right, let's look at, I don't know. This is color, and this is presence of color. Right, so white cats are entirely white, right? And if you cross these together and you get your heterozygotes, you'll get baby cat, who's a little chunkier because I'm drawing it funny, will also be white. because white is dominant over any color, right? So this is a classic example of dominant epistasis. And so if you have a dominant Y allele and you only need one of them, you will never see the genotype that you have for the color gene. So you will get, how many will that be? That's a 12:3:1 ratio. There you go. So essentially you'll get either white, which have a dominant Y allele. You'll get black, which are homozygous recessive and have a black allele, right? Black being pigment production. And you'll have orange, which are homozygous recessive for both. And that's in a 12:3:1 ratio. And so this is called dominant epistasis because it masks the phenotype at another locus simply by having one functional dominant allele. And the reason for that is that this is all about melanin production in cats. And so if you have one dominant W allele, you have no melanin production. So it doesn't matter whether you're going to be producing dark melanin to have black cats or light melanin to have, or no melanin to produce ginger cats, you'll end up with white cats. There's just no melanin production whatsoever. So that's not quite right. It's not no melanin for ginger cats, it's a different type of melanin. One that blocks less color. Right, so this is a different case of epistasis, and there are lots of different types of epistasis, depending on essentially how those different alleles interact. You can have ones where you have two different pathways, right, which complement each other, and so you get a different outcome. So essentially, the key characteristic of epistasis is a non-Mendelian F2 generation and dihybrid cross. So normally it's 9:3:1. Epistasis is all kinds of different numbers. Could be 9:7 or 12:3:1. Oops. Or another whole number. That's pretty much whole numbers, right? So one thing that I want you to be careful about is you can get other deviations from that 9:3:1 ratio. But what you would the other deviation would be linkage, right? And so that would be, how can I put that? Is essentially linkage isn't that, right? Linkage is essentially kind of like linkage would be if you see fewer of those non-parental phenotype combinations, but you still see them. Epistasis, typically you will not see one of either you won't see one of the parental phenotype combinations because it's masked, as in the case of dominant epistasis, or if you're looking at a 2-gene like dihybrid cross, you will not see 4 different phenotypes. That's the case of the flower pigment that we just looked at, the recessive epistasis. Right. So if in the essentially in the F2 generation you have less than 4 different things, it's epistasis. If you have 4 different phenotype combinations, but the ratio doesn't match, then it's probably linkage. That's probably the simplest way of kind of saying that, I think. But linkage will make a lot more sense once we've spent our 2 lectures at least going through it. I'd strongly recommend you look through that PowerPoint before we hit it on Thursday. Linkage is something that students really struggle with, right? Because there's a lot of math involved. There's a lot of kind of changing how you think about stuff, essentially. So if math isn't your strong suit, check out that PowerPoint. It'll look just like this. Ta-da! This is what we'll be going over on Thursday. Okay, so ever so quickly, right, we've got, oh shit bits, kind of running out of time. Okay, spent a little bit too long on that. So ever so quickly, there's this concept of complementation, right? And so this is really more of a technique in terms of genetics, right? So if you have 2 different mutations with similar phenotypes, you want to figure out, are these alleles of the same gene or different genes? Then what you do is what's called a complementation assay, right? And so if you have homozygous recessive, well, essentially homozygous recessive of each of those different alleles, right? So A and B here, and you cross them together and you still see the mutant phenotype, is because essentially you've put one mutant allele with another mutant allele, right? And so you still don't have function. I don't know, somewhere else, love. That's cool. Actually, this is technically this is a heterozygote, right? But this doesn't have the dominant phenotype because it's a compound heterozygote. It's 2 recessive alleles together. Now, if these mutant alleles are in different genes, right? Different parts of chromosome or even different chromosomes, then if you cross those together, they will complement each other, because essentially what you're doing is you're taking one mutant allele from one and adding it to a wild type allele from another, right? Because if it's different genes, then in one mutant you'll have homozygous recessive mutant for one gene, and the rest will be normal, right? So for the second gene, you'll have wild type dominant alleles, and it'll be the other way around for the other mutant. And so when you cross those together, you're going to get heterozygotes for both. And so you'll see function, and then you'll have the wild type phenotype, right? And so this is just really, if you isolate, like you do a mutant screen, or you throw a load of nasties on worms and you get a whole bunch of mutants out, and they all have the same phenotype, this is the first thing that you do to figure out whether or not those alleles that you created are in the same gene or in different genes, right? And they could well be in different genes, right? If that phenotype is a result of, again, that kind of biochemical pathway that you saw, then you could even do an epistasis experiment to see which order they're in, for example. I'm not going to really talk about sex influence traits, and don't worry, because that's not going to be in the exam. This is kind of stuff that's left over from the previous person that taught it. What we do need to talk about is quickly variable expressivity. Actually, if we run over, we'll just can pick this up on Thursday, so we don't have to rush it. 2 other concepts, right? Variable expressivity and variable penetrance. And so the idea here is that you have the same genotype as another individual, but a different phenotype. Not necessarily, actually, no, that's not correct. Not a different phenotype, a different degree to which you express that phenotype, right? So the phenotype would be the same, but you may suffer a more severe form of that disease compared to another individual that has the same genotype. Or you might not even have the disease at all, right? Still with the same genotype. So obviously this is where things get really kind of complex. And if you express it to a different degree, that's called variable expressivity, which kind of makes sense. And if you have cases where you have that genotype, but that phenotype is not expressed, that's called incomplete penetrance. So that basically means that not all individuals in a population with that genotype have that phenotype. That phenotype hasn't penetrated the population completely. So I have a really good example that I like to talk about, which is actually kind of talks about both, right? And so how many of you have heard of mitochondrial disease? Just out of curiosity. Is this something that's kind of crossed your radar so far? It's kind of an interesting one. It was often dismissed as being kind of like a yuppie flu. I don't know if any of you even remember what the word yuppie means, right? But kind of like people who get a little bit tired more easily than others. It's just basically kind of blown off as being you're just kind of making this stuff up type thing. So in your cell, obviously, you have mitochondria. Mitochondria also have their own genome, a limited one. So a circular genome, which they essentially is a relic from when they were prokaryotes, right? Before they kind of got absorbed by our long ago ancestors. And those genes are all involved in mitochondrial function. Typically, genes involved in electron transport chain or oxidative phosphorylation, stuff like that. Now, not all of those genes aren't all of the function of mitochondria. There's a lot of function that comes from nuclear genes, right, which you inherit from both your mother and your father. But there are a subset of those functions which come just from the genome of the mitochondria itself, right? And that genome you inherit from your mother only, right? It's a matrilineal. I can trace my mitochondrial DNA back, potentially tens of thousands of years if I wish to, through my mother and her mother and her mother and her mother and her mother and so on, right? Which is kind of neat. It's kind of the opposite of that Y chromosome haplotype that we talked about last lecture. Anyway, so the issue is if you have mutations in those mitochondrial genes, right? And so this is where it becomes really complex, right? So mitochondria are haploid, right? They only have one copy of each gene, right? Compared to our nuclear DNA, which has two copies because we're diploid. But you have more than one mitochondria in each cell. You actually can have, hundreds, right? Depending on the type of cell that we're talking about. Muscle cells, for example, are almost entirely mitochondria and actomyosin filaments. Whereas things like adipose cells, that fat storage cells have very few, right? Because they don't really do anything. So so if you inherit one of these mutations from your mother, right, it depends on how many of the mitochondria in your cell have that mutation, right? Because it's a population. It's like a population of bacteria in a way, kind of inside your every one of your cells, and also what's needed of that cell itself, right? And so with mitochondrial disease, you have this huge spectrum of disease ranging from, you know, just feeling a bit tired after a long day's work, right? Or go out for a walk and you kind of get kind of more fatigued than you should do, all the way to not making it out of childhood, right? Dying as an individual, right? And everything in between, right? So this is the same genotype, but you have this huge range in expression of that phenotype, right? Ranging from people who can live very normal lives to people who die before they reach the age of 3 or 4, right? And also it depends on where you have those mutant mitochondria, right? Because you inherit a population from your mother. They're not clonal, they're not all the same. So if you have mutant mitochondria more in your neurons than elsewhere, then you're more likely to have mental retardation or kind of cognitive and motor problems, right? If you have it in your muscles, then you're likely to get heavily fatigued or be unable to exercise for long, right? Because you don't have that ATP function, right? So that, depending on where those mutant mitochondria are, depends on what kind of phenotype you express as well, and to which level. So the whole penetrance thing is kind of another take on that, right? So this is why I like talking about this. So if you have sufficient number of mitochondria in your cells to provide function for that cell, you will not see the mutant genotype of the dysfunctional mitochondria. So now we have kind of essentially a threshold effect, right? Where you can have kind of a number of mutant mitochondria below that threshold and you still have function, right? However, once the number of mutant mitochondria breaches that threshold, now you show the disease phenotype, and to a different degree dependent on the number of the relative number of mutant mitochondria, right? So now you have essentially variable penetrance depending on, you know, which tissue we're talking about, which individual we're talking about, which level of mutant mitochondria we're talking about, right? So mitochondrial disease is a fabulous example of kind of the complexity, right, of some particular traits, right? Some individuals can be completely fine. You'll never even know that you have mutations in your mitochondrial DNA. Other individuals, like there's a woman in the UK who she was actually fine. Her I don't think she really expressed if any of this, but she had a sufficient kind of load of mitochondrial mutations or dysfunctional mitochondria that every single one of her children died before reaching adulthood. I think she had 7 children in total, and unfortunately not one of them survived. So she was actually very important in the drive to create what are called 3-parent babies. I don't know if you've ever heard of that. It's kind of a fun thing to talk about. So essentially, she couldn't produce children that would survive because every one of her oocytes that she has with which to create children have mutant mitochondria in them. And so it's now legal. I don't think it is in the US. It's for various reasons, but in the UK you can now essentially take a sperm and an egg, fuse them together, right? So you have a fertilized zygote. So that's your nuclear and nuclear DNA. Stick those together. Then you take that nucleus out of that fertilized zygote and you put that into another oocyte from another woman who's had the nuclear DNA taken out. And so essentially that oocyte from the other donor has healthy mitochondria. And then you're taking the two versions of your two lots of chromosomes and you're sticking it into that cell. And so that individual, when they, become an individual, will technically have three parents. It will have a mother, a father, and a mitochondrial mother. And then that will break that transmission of those disease mitochondria. Anyway, so we didn't get time to talk about complex traits. We'll do that beginning of next class because that's something. It won't take very long. It's kind of talk about in like broad, fluffy terms by and large, but we'll talk about that at the beginning of next class. In the meantime, please do look through the linkage PowerPoint so that you're not completely blown up by what we talk about on Thursday. All right, that's it from me. If anybody has any questions, feel free to drop them off. In the meantime, I shall, oh, hang on, crap, sorry, Rose, I didn't see that question. Yeah, so if you carry the genotype for a disease, let's say a homozygous recessive genotype, which normally would result in disease, but you don't have it, that disease would said to have incomplete penetrance, right? Because even though you have the genotype, you don't express the phenotype. And that's typically because of other things going on, right, that influence the expression of that genotype. Yeah, so that's kind of what incomplete penetrance means, is essentially there's something else which can either maybe supplement that function or kind of get around that issue in another way so you don't present that that disease phenotype. So again, that's kind of leading really in a way into complex traits. Cool. All right. Thanks very much, everybody. I shall see you, if not tomorrow, and I shall see you on Thursday.